understanding neuronal communication and synaptic plasticity. Starting on the far left, we have dendrites, also termed dendritic spines. These are the ears of the neuron, where information from other neurons flows in. Sitting next to the dendrite is the cell body, a basic part of every cell. Moving right, we reach the axon, which communicates to the next neuron, terminating in axon terminals. When the neuron has something to say, a wave of excitation starts at the dendrites, spreading over the cell body to the axon, reaching the axon hillock, and shooting down the axon to the axon terminals, then to the next neuron. The nervous system is quiet when there's no message, known as the resting potential. Neurons work to keep ions separated, using energy to maintain this state. An ionic change at the dendrites switches them from a resting to an excited state, causing a wave of ionic change that sweeps down to the axon. This excitation, starting at a dendritic spine, can be measured with an electrode. The wave of excitation decreases over distance, so summation is needed, either temporal, one neuron firing repeatedly, or spatial, many neurons firing together, to reach the axon hillock and pass the signal. The axon hillock decides whether to pass the signal based on the wave of excitation. If the threshold is reached, an action potential occurs, shooting down the axon without losing force. The axon hillock changes its threshold over time due to hormones affecting neuron excitability. Neurons communicate through synapses, gaps filled with extracellular fluid. Electrical signals can't cross synapses directly. They convert to chemical signals. Neurotransmitters in the presynaptic neuron are released into the synapse, affecting the postsynaptic neuron. Different neurotransmitters have varying excitatory or inhibitory effects. Learning involves changes in synaptic communication, and memory involves long-lasting changes. The adult brain forms new neurons and synapses, but strengthening pre-existing synapses remains the dominant learning mechanism. This concept by Donald Hebb involves potentiating synapses, making communication more likely. Long-term potentiation, LTP, is the process where repeated stimulation makes synapses more excitable, critical in learning and memory. LTP involves repeated stimulation, causing a greater wave of excitation. For example, stimulating the amygdala can condition a phobia or PTSD through a neurotransmitter called glutamate, the most excitatory neurotransmitter. Glutamate has two types of receptors, with the second activated only by a large amount of glutamate, allowing calcium influx, potentiating the pathway. Pre-LTP synapses have neurotransmitters in the presynaptic neuron and receptors in the postsynaptic neuron. Calcium initiates pathways, pulling spare receptors onto the surface, increasing postsynaptic sensitivity. Receptors undergo phosphorylation, altering their function. Dendritic spines change shape, with more receptors staying open longer, enhancing excitation. Retrograde neurotransmitters signal the presynaptic neuron to release more glutamate, reinforcing the signal. This is what LTP and potentiated synapses are about, crucial for understanding learning and memory.